Hello everyone. Welcome to my YouTube channel. Today this is my first session of my YouTube, and where we will discuss about the differential expression of genes in cancer. Okay, for this we will understand how the gene activity changes driving the tumor biology. And for this, the topics will be covered. Number one, the what is DIG. Number two, why it matters. And after that, how it's done actually. Number four, the methods overview. Number five, the clinical applications of this DG gene identification. Okay, so in the coming to the next slide, where here the differential gene expression definition is given. Differential gene expression refers to the significant gene differences. Significant difference means it should be mathematically and logically correct. Means it should be statistically correct. Okay. For this is significant difference in mRNA protein levels of a gene between two or more logical conditions, such as cancerous versus normal tissue. Two or more. Why it is written like that? Because if some genes are there, gene I'm thinking like gene X, then gene X expression in the cancer, then cancer stages: stage 1, stage 2, stage 3, stage 4, like this. Then these four stages, how the cancer expression is there, we have to identify. Okay, and now we'll see in this diagrams where the expression profiling in cancer and normal is given for some genes: gene A, gene B, then gene C, then gene D. Where in case of gene A, the expression is significantly high. I am thinking like that because the blue one is the cancer and the orange one is the normal. So here we can easily see that the cancer having high gene expression than the normal. But in case of gene B, it is fully different. The cancer having the gene B expression is lower than the normal. Okay. So normal has a high expression of these genes. And in case of gene C, like the gene A only, and in case of gene D, it is upregulated than the normal. Okay. Then can we take upregulated, downregulated? You can understand from there. Then gene A, gene C, gene D. These three genes are upregulated genes in cancer. And in case of gene B, this is the downregulated gene in cancer. But here the upregulated genes and downregulated genes are there. So can we tell them as differentially expressed genes? If yes, then how? And if not, then why? So for this answer, there I have written here generally. Generally, it is used as for the differential expression of genes in case of statistical models or statistical methods are there. So for this, the fold change greater equals to 2 and adjacent p-value less than 0.05. Fold change means what is this? Fold change means nothing. Like gene A is there, fold change is equals to then gene A expression in the cancer, mean expression, mean value of the gene A in the expression of the cancer patients by mean expression in the normal patients of the gene A only. So this is a fold change. If it's greater than 2, greater equals to 2, then it will be selected, or if not, then it will not selected. You understand from this? If this is, then it is okay. Then if we can told them, we can tell them as the differentially expressed genes, and if not, then we cannot. Okay. And here the adjacent p value should be less than 0.05, where it can be taken as 0. Less than 0.01 also for some time when the gene number is so much. Okay. So in case of upregulated genes, what you know easily that upregulated genes, what is upregulated genes like this? The in case of upregulated genes, the MEK, EGFR, KRAS genes are there. And in case of downregulated genes, TP53, BRCA1, P10 genes are there. And I am thinking you as a biologist, so you know what is TP53 gene or the function of it, and how it increasing, how its downregulation is increasing in case of cancer patients. You should know, but I am telling, as for example, I am telling that. Okay, I am just giving the overview of the differential expression genes. For this, I am telling you, we will discuss about the downregulated genes or upregulated genes in later, but you should know some basics that upregulated genes means overexpression in case of cancer than the normal, and downregulated genes means the cancer patients having less expression of the genes than the normal. Okay, that is the scenario of the gene A and gene B. The gene A is the upregulated genes and gene B is the downregulation genes. Okay, so here in the next in this slide, we have to know why is DEG analysis is important. Okay, in number 1 point is biomarker discovery. What is biomarker? Biomarker means the markers present in the biological system. Biological system means if I am telling about the human body or in the cellular level or in the molecular level, if the markers expression level, we can know from there we can predict the disease is there or not. If the glucose level is high, then we can tell, generally we are telling like that high glucose level or high sugar patients, and generally we are telling like that if the level certain level below the certain level, then we can tell as low sugar patients, that we are generally telling na, that there the glucose is a biomarker. So from there you can understand, but here we are giving some example like PSA in prostate cancer. PSA means prostate specific antigen level can tell the cancer is there or not in case of prostate cancer patients. So, and in case of breast cancer, the HER2/neu is a potent biomarker for the HER2/neu subtype, third subtype of the breast cancer mainly. Number 2 point, the drug target identification. If we are understanding the DEG genes, then differentially expressed genes, and based on their expression level changes from the normal level, what pathways they are modulating, we have to understand. If the pathways we understood, then we can use certain drugs that can be inhibited that pathway related genes or proteins like EGFR to inhibit the EGFR, then EGFR inhibitors. To inhibit the BRAF proteins, the BRAF inhibitor drugs are there. Number 3 point, molecular subtype. This molecular subtyping means classifies the tumor by gene expression profiles, enabling the personalized treatment decisions. Example, BAM50 subtypes in breast cancer. Like if we have the expression level of the HER2/neu, if I'm telling his example, HER2/neu is the potential biomarker for the HER2/neu subtype. So here in case of luminal A, luminal B, HER2/neu, and for the triple negative breast cancer, these are the four most important subtypes in case of breast cancer. Okay, in case of luminal subtypes, the estrogen and progesterone receptor expression is prevalent. And in case of HER2/neu, the HER2 receptor, and in case of triple negative breast cancer, these three are not there. Is estrogen negative, progesterone negative, and HER2 negative. So based on this certain receptor or protein level expression, we are predicting the subtypes mostly. But there are certain more genes are there or protein expression are there based on which we can also use that. But this is the potential ones. These are the okay. Then pathway analysis. I already told you, if we know the dysregulated or differentially expressed genes, then we can also predict from there which expression, which genes are related to which pathways, and by the expression changes, dysregulating the expression level in case of cancers, which pathways are modulated, inhibited, or upregulated by the gene expressions. And if we predict that, this will directly useful in case of drug target identification. Number 5, the prognostic prediction. The genes we have already identified, we can study the genes and we can predict that how the gene is responsible for the survival of the patients. Okay, it is statistically significant or not, we can predict. And for this, we are generally using some famous Kaplan-Meier plot. KM plot is there. Okay, when the p-value is less than 0.05, p-value means log rank p-value. We'll study this definitely. Then precision oncology. Precision oncology means the patient specific or some gene specific way, because if in some patients some gene expression, means gene A, gene B expression is suddenly increased and their survival is decreased, we can give the certain types of the patient group of subtypes, group of patients, that group of patients, we can give some inhibitors specifically for the precision oncology study for inhibiting the disease progression. That is just a basic thing in case of precision oncology, but it is a very big chapter for the translational biology or for the genomics study of biology. And how do we perform the DEG analysis? Here, at first, the samples should be collected from the clinics or the healthcare centers. And after their RNA extraction from the cultured cells from the samples, raw samples, and then quality control means methods are there. We have to follow that. And again, for the sequencing or microarray RNA sequencing or microarray data, we can generate from there, followed by the pre-processing steps where the align reads like star, he said to count reads feature, then PMM normalization methods are there for the pre-processing steps. Okay, again, for the statistical testing, we will go for differentially expression genes, and where we'll apply D-SIC2, AGR, and limma for the R path, R languages, R studio apps, and for Python app in Jupyter Notebook, we have what we'll discuss in the Jupyter Notebook, how the Python package is also used. We will see in case of statistical testing, and for the filtration, we'll apply the FC greater than 2, FC greater equals to 2, and FDR less than 0.05 cutoffs, as where sometimes I already told that less than 0.01 also taking when the gene number, differentially expressed genes number are so much is coming from the less than 0.05 cutoff. Okay, so after that, perform the GAC or pathway enrichment analysis to see the pathway are linked to this differentially expressed genes expression analysis. And that is that. And in the next slide, in case of key methods and tools, there are 4 types of data: RNA sequence, microarray, scRNA sequence, spatial transcriptomics. Like this data are there. Actually, the RNA sequence is the whole transcriptome sequencing, and microarray is the probes for known gene sequences, and scRNA profiles each cell individually, the cancer and adjacent normal tissue. So like this happens in the single cell RNA sequencing data. It is advanced, so much advanced, and in nowadays the spatial transcriptome is so much increasing nowadays, and it is evolution by evolution. At first, we'll see the evolution also. Actually, at first 1990 1995 to 2000, the microarray era, the microarray happens, where the first the breast cancer subtypes for this, the microarray profiling happens. And after the 2008 to 2012, the RNA sequence revolution, next generation sequencing are there. Again, after 2013 to 2018, the pathway and network biology, network analysis happens, and how the gene level networking happens will we seen that time. Okay, after this the revolution happened that time. Okay, nowadays also you are also using some smart methods are also there, the AI also inbuilt there, and nowadays it is happening. And 2018 to 2022, single cell DG genes, and 2022 to now the spatial transcriptomics data analysis was occurring. So it is revoluting like this. Okay, DG genes in cancer research. And after what steps I told you, then after that, in case of key visualization in DG genes, this type of volcano plot will see where the downregulated genes are in the blue color and the upregulated genes in the red color. Okay, these genes are red color genes are the upregulated genes, and downregulated genes are in the blue color, and in between that the insignificant genes. Okay, that genes, the gray colored genes are insignificant genes. Okay, and here from here to here like this, it is the 1 plus 1, and here -1 to -15, it is the downregulated genes. So log to FC value greater than 1 is for the upregulated genes, and log to FC value less than -1, it's for the downregulated genes. We are taking like that, and -1 to 1, this distance -1 to 1, these are these genes are insignificant genes, and here the log 10 adjacent p-value also in the y-axis for the statistical significance. Okay, and for the FC mean expression value of cancer patients, like this, the mean expression value of normal patients, and this is there by normal patients. And for log 2 FC value, log 2 is equals to 1, and if FC greater than 2, this is the calculation you can easily see from here. Okay, so this is the case, and also there are some plots are there, like heat map also there, heat maps are there, and then MA plot, and the PCA, principal component analysis plot also there, and the TSNE, UMAP, like these plots also there. For the GACA and pathway enrichment analysis, the bar or bubble plots also there for the identifying the which pathway are dysregulating by or after the dysregulation of this certain genes, and we have to identify that, and this is type of that type of scenario will be happen. In case of the clinical applications of DEG in cancer, that first the example TCGA project. The TCGA project is there, means TCGA is a data repository where 33 types of cancers with the 11,000 tumor sample data is there with the normals also. And here they make the data available for the public. Okay, it is the public data repository. We can easily use that, but there is some procedure I will show you later, how to download the data and how to go for the data analysis from this data. Okay, and after that, the tumor microenvironment. The DEG identifies immune cell infiltration signatures like trimer, cybersort to predict the immunotherapy response in melanoma then NSCLC, the subtype of the lung cancer, and the bladder cancers are there, certain bladder cancer that they are predicting like that. And for the oncotype DX, a 21-year expression assay for early stage breast cancer, then DEG, derived gene signatures, predicts 10-year recurrence risk, guides chemotherapy decisions. Then drug resistance pattern, the comparing the pre and post treatment transcriptome analysis, the drug resistance also we can predict it from there, because every drug cannot affect on every cancers, or the same cancer subtype is there, but the patients are different. Then also every patient is not effectful for the same cancer dose or cancer drug. Okay, that is so much critical matter. I understand you are thinking of that, but we will discuss about the latter, how we can predict that and how we can do that. The drug resistance we can predict from the data and from the study of the wet lab also, and how we can remove that by the another drug, and we will predict about that. Okay, what we just discussed about the differential gene expression in cancer. Okay, in number one, the DEG measures the significant differences in the gene expression between the cancer and normal tissue, revealing up or downregulated genes. Okay, in case of number two, the critical for discovering the biomarkers, drug target, tumor subtypes, and predicting the patient outcomes in precision oncology. Number three, the workflow. At first, the sample you have to take. After that, from there, the RNA sequencing or microarray data sequencing happen, and then normalization of the data and statistical testing like D-seq2 or AGR for the in the R cases, R language, and after that the visualization and pathway analysis should be done. Number four, the evolved microarrays to bulk RNA and then single cell, then spatial transcriptomics nowadays is so much evolving nowadays. So in the next time, thus we'll understand then at first the bulk RNA, then single cell, then spatial. Okay, we will talk about that. And then number five, the real world impact, the TCGA, Atlas, Oncotype DX assay, etc. Then how the immunotherapy response prediction and the drug resistance is happening in nowadays for the cancer patients, and how we can prohibit that, the way of that we have to identify. In the next class, we'll discuss about the statistical test for DEG gene analysis, what is the different methods are there, and what is the basics and mathematical background of this all, and how we can use the Python code and R code in this. I will share the video one by one, no problem is there, just to study. And if you have any doubt, then post in the comment section. And okay, bye. Thank you.